Okay, well, I'd like to welcome everyone to the second of our COVID lockdown clinical and translational research grand rounds. And it's a pleasure for us at the Georgetown University Center for Clinical and Translational Science and MedStar Health Research Institute to welcome everyone to hopefully a stimulating 1 hour break from what's otherwise either a clinical onslaught or a frustrating misery of these months. So our speaker this month is Scott Turner, who's professor of neurology and director of the memory disorders program at Georgetown. His clinical and translational research, at least when he gets to return to doing it, focuses on Alzheimer's disease and related dementias, including prevention, diagnosis, prediction, and treatment strategies. Using tools such as imaging, biomarker studies, studies of mechanism, and as you'll see, randomized clinical trials. And he's going to present some research findings, including results of a randomized trial of nilotinib for Alzheimer's disease. Scott, ever the consummate clinical investigator, is the only one of our speakers who asked to use the space in the announcement for his talk as a recruiting tool, which you all saw. And so with that, Scott.
Thank you for the invitation and welcome everyone. I think we can probably skip through these introductory slides. Here's the text number for CME. And these are the objectives. We're going to talk a little bit about background of Alzheimer's disease, including the new biomarkers that we've developed, and talk about therapeutic strategies. And I'll talk a little bit about our recently completed phase 2 clinical trial of nilotinib for mild Alzheimer's disease, which has now been resubmitted for publication. These are the disclosures. We get funding for our research from federal sources, mostly National Institute on Aging, foundations, Alzheimer's Drug Discovery Foundation in particular, and we do multi-center studies sponsored by various corporations, and we also get some support from philanthropy. This is the Memory Disorders Program, is myself, for nurse practitioners. Thank you to all the members of the Memory Disorders Program, and thank you also to the Clinical Research Unit. We couldn't do all of our studies without you. We've been doing many, many studies, as you know, over the past 12 years since I came to Georgetown. These are the drugs that are approved for Alzheimer's disease, so these are very old clinical trials. And there are 3 cholinesterase inhibitors and 1 NMDA receptor antagonist. You can see that the most recent drug was approved in 2003, meaning that all the phase 3 clinical trials that have been conducted since then have not been successful. So that's been a big disappointment in our field, obviously. This was mostly a marketing move in 2014, combining 2 drugs as they were going off patent. So we typically recommend prescribe one of these 3 cholinesterase inhibitors once you're diagnosed with Alzheimer's disease, mild Alzheimer's. And then when you get to the moderate stage, we add memantine, is how they are approved and recommended, and that's the evidence-based practice. So since we've had a lot of failures since 2003, we're starting to do things differently, of course. So we traditionally focused on dementia, but now we're focusing more on the MCI, mild cognitive impairment subjects. And now we're also moving into the preclinical stage of Alzheimer's. And the trick is to discover those people who are going on the solid line trajectory compared to those going on the dotted line trajectory. And this is where the biomarkers come in that I'll be showing you and talking about to identify these people on the dotted line from these people on the solid line. So we're doing more and more research of MCI in preclinical stages, and here we're getting into the prevention trials. We're still doing some clinical trials of mild to moderate Alzheimer's, and once you have dementia, we haven't given up on the dementia group, but we're doing more MCI and preclinical trials in addition to dementia. What have we learned in the last 20, 30 years? We now know here's the amyloid plaques, the neurofibrillary tangles. The plaques are mostly made of the A-beta peptide that comes from APP by proteases called BACE1, beta amyloid cleaving enzyme 1, and gamma secretase. And these release the APP as a byproduct of APP metabolism. This accumulates and precipitates, changes conformation into these large amyloid plaques that you see on the silver stain of these brain sections. This is neurotoxic, promotes a lot of other pathologies, including gliosis and inflammation. This is the neurofibrillary tangle made up mostly of phospho-tau protein. So there are two obvious drug targets on this slide. One is BACE1 and another is gamma secretase. And we've done a lot of work and a lot of clinical trials testing different BACE1 inhibitors and gamma secretase inhibitors. And we know that they're successful in hitting the target, but they have not been successful clinically, and these strategies are basically gone back to the lab for the most part because they don't seem to be effective, and in fact, they may be making cognition perhaps even worse compared to placebo, and that's probably because of off-target effects. These proteases are cleaving a lot of other proteins other than APP, and so we're running into problems. Oh, CME code 16855. We've also learned a lot about genetics. So there are 3 autosomal dominant mutations in APP and presenilin 1, presenilin 2. The presenilins are actually part of the gamma secretase complex. These are very rare in the population, about 100 pedigrees in the world. But they have 100% penetrance, and different pedigrees have different age of onset, 20s, 30s, 40s. So if you do have this autosomal dominant mutation, you will get Alzheimer's very assuredly if you live to that age. And there are also hundreds of genes, gene variants that affect Alzheimer's risk that are very common but have very, very low impact on your risk. But there are lots of genes in this category. And then there's the ApoE genes, ApoE4 being the high-risk gene. 2 copies is, 1 copy is 3 times the risk, 2 copies is 8 times the risk. And then more recently, there's a genetic variant of TREM2, a receptor on myeloid cells, in this case in microglial cells, which also influences risk of Alzheimer's and clearly points to inflammation as part of the pathology of Alzheimer's, and inflammation, anti-inflammatories as a potential therapeutic strategy. So again, here's the beta secretase and gamma secretase cutting A beta, and then this aggregating and forming amyloid plaques. We have not got any traction in BACE1 and gamma secretase inhibitors, so I'm going to talk more about degradation and clearance. And one way to promote degradation and clearance is by immunotherapies, which can be active, injecting the A beta as the antigen, or passive, infusing anti-A beta monoclonal antibodies, where we have got more traction. And then I'll talk about a new strategy of intracellular degradation of A beta, targeting tyrosine kinase inhibitors with nilotinib. So again, here's the intracellular A beta released from the neurons. Dimers, trimers, oligomers, all of these are neurotoxic, promote inflammation from the glial cells, and promote tau and tangle formation. And here are the larger amyloid plaques that you see on silver stain. And these are the ones you see on amyloid PET scans as well. So you don't really see these forms of the A beta either by silver stain of brain sections or by amyloid PET, but we think that they're also equally neurotoxic. To see these, you need to do immunostains of Alzheimer's brain. So we've formed this amyloid cascade hypothesis based on the genetic evidence, these familial mutations that promote A beta generation from APP. Down syndrome is another risk factor, trisomy 21, where APP lives on chromosome 21. ApoE4 is a risk factor. Number 1 risk factor is aging. APP turnover generates A beta that aggregates, is neurotoxic, promotes tangles. You get mild cognitive impairment, inflammation, encephalopathy as seen on a glucose PET scan. the neuronal morbidity and mortality, white matter changes on MRI, dementia and death, and this takes 20, 30 years. These are our current drug treatments. They're relatively downstream, so they have modest palliative benefits. These are the biomarkers and when they become abnormal: low A beta and high tau in spinal fluid, positive amyloid PET scan, abnormal glucose metabolism on PET, and then atrophy and white matter changes on MRI. The best biomarkers so far, the best evidence, is CSF A beta 42 and less A beta 40, PET amyloid imaging, CSF tau and phospho-tau, hippocampal volume on MRI scan, rate of atrophy on MRI scan, glucose PET, and there are lots of others in the running, including a possible blood test for Alzheimer's, which still needs further work, not quite ready for prime time yet. And these are the famous Jack curves of when the biomarkers become abnormal. So here you go, preclinical Alzheimer's to mild cognitive impairment. This dotted line means that you begin to have significant cognitive deficits that deviate from the mean for someone of your age and education. And this dotted line means you not only have cognitive deficits, but also functional deficits, which is how we define dementia. So this is functional decline, this is cognitive decline. And the first biomarkers that become abnormal are the amyloid in the spinal fluid and amyloid PET scan, followed by tau and glucose, and then the volumetric MRI. You can also see there's a ceiling effect, so that there's very little correlation of the amount of amyloid in the brain and with cognition. And this is how we define the preclinical Alzheimer's individuals. For example, someone who has an abnormal A beta and tau in CSF or a positive amyloid PET scan, we know that they're on track to eventually cross into the MCI and dementia stage. This is what the volumetric MRI looks like. Everything in blue is parts of the brain that are shrinking about 1% per year between annual MRI scans. Parts in the light blue are shrinking 3% per year. This is the glucose PET scan. This is temporoparietal hypometabolism, posterior cingulate, cuneate hypometabolism in an Alzheimer patient. You also see milder deficits in a mild cognitive impairment patient. So this is an abnormal glucose PET, and this is the focal encephalopathy that I was talking about in the amyloid cascade hypothesis. This is a part of the brain is not functioning and therefore not taking up glucose because it's diseased by Alzheimer's pathologies, including A beta, amyloid, tau pathologies, and neuroinflammation. So we can get a glucose PET clinically, and this would be a pattern that would be diagnostic of Alzheimer's. Another biomarker is proteins, proteomics in spinal fluid. So here's an elevated tau and low A beta in Alzheimer patients compared to normal individuals. You can also look at other proteins as well, many other proteins also in the running. But this is relatively the new technology, which is the amyloid PET scans developed in the last 10 years or so. And then more recently, tau PET scans developed in the last 3 to 5 years or so. There are now 3 FDA-approved amyloid PET ligands. There are no FDA-approved tau PET ligands as yet, but they're coming. And this is the amyloid uptake, amyloid binding in a clinically normal individual, another clinically normal individual, and then a patient with Alzheimer's disease. So you begin to see this medial temporal hippocampal amyloid uptake spreading throughout the cortex as you go from normal to Alzheimer's. And then again, medial temporal hippocampal uptake of tau tangles by the tau PET scan, also progressing and accumulating, getting worse as you go from normal to Alzheimer's. So I'll talk about one of the trials, the anti-amyloid monoclonal antibodies, which is called Aducanumab. MAB stands for monoclonal antibody. This is a Biogen antibody. We did the phase 2 trial at Georgetown in the CRU, and we also did a phase 3 trial at Georgetown in the CRU. This is a multi-center international studies. This is the amyloid PET scan at baseline. And this is the amyloid PET scan 1 year later of individuals given placebo, 3, 6, or 10 mg per kg IV infusion monthly of Aducanumab. And you can see that there's a dose-dependent reduction in amyloid in the brain as a result of the anti-amyloid antibodies. So it's very clear that these antibodies can remove amyloid plaque from the brain And you can go from a positive amyloid PET scan to a negative amyloid PET scan as a result of the Aducanumab treatment. And this can be quantified. Here's the placebo group versus this dose-dependent reduction in the SUVR, the amyloid burden in Alzheimer patients. So reduction in amyloid burden as a result of the antibody treatment. The harder question is, does removing the amyloid have a beneficial effect? And at week 52, here's the decline seen in the placebo group as expected, as anticipated. Reduction of about 2.5 points in a year in the placebo group, and less reduction with somewhat dose dependence of the Aducanumab stabilizing cognition, meaning that these individuals who got the highest dose did not decline clinically in their MMSE. So this was results of the phase 2 data, which were very promising, obviously. And so phase 3 studies were launched, but I'll just mention first the major side effect of these immunotherapies, and that's ARIA-E and ARIA-H. ARIA meaning amyloid-related imaging abnormality edema, amyloid-related imaging abnormality hemorrhagic. So for example, here was one patient that we had enrolled in the study at Georgetown. MMSE scores were fairly stable, so we found out that he was getting placebo during this double-blind phase of the study. Then when we switched to the open label where he got the real medication, there was a precipitous decline in the MMSE from 26 to 11. At this point, drug was withheld and he slowly returned to baseline over 2 to 3 months. So what happened to this individual? This was the MRI scan at baseline. This was the MRI scan at his worst. You can see all of this ARIA-E, all of this edema in the MRI scan here posteriorly, and a small hemorrhagic stroke as well. So this individual had both ARIA-E and ARIA-H as a side effect of his Aducanumab treatment. And then it was discontinued, and a few months later, mostly resolution. There's still some blood products here at the hemorrhage. So mostly resolution and return to baseline as a result of this ARIA. So this is the major worrisome, troubling, dose-limiting side effect of these immunotherapies for Alzheimer's disease. So based on the promising phase 2 clinical trials, 2 phase 3 clinical trials were proposed and conducted. And the reason 2 phase 3 trials were proposed is because this is what is required by the FDA for a new drug approval. We need, the FDA requires 2 phase 3 clinical trials with positive results, meeting the primary endpoint, pre-specified endpoint of the clinical trials. And so they were very similar in structure, Engage and Emerge. They were international, multi-center studies. I think Engage launched a little bit before Emerge, and but otherwise they were very similar. But in March of last year, both of these trials were discontinued because of a futility analysis suggesting that these drugs were not efficacious. But while they were analyzing this data, they were also collecting more data between December and March. And then when they collected further data in October, they announced, there was a positive outcome in one of the trials, which met the primary outcome. And the other trial was not positive, but if you look at a subgroup that got the highest dose for the longest time, it also seemed to be supportive of a positive outcome. The major side effect was ARIA-E in 35%. Most of it was asymptomatic, meaning it was detectable only by MRI scan, but no signs or symptoms. If it was symptomatic, headache was the #1 symptom in 20%. Based on this data, the company, Biogen, is submitting a new drug application to the FDA. It was supposed to be submitted this summer, but it's been delayed somewhat, so that it will be submitted probably in the fall of 2020, and the FDA will make some sort of decision about this drug. So let me show you some of the data so you can make up your own mind about whether you think the FDA should approve this drug or not. So here is the result of the Emerge phase 3 clinical trial. So you can see each group had more than 500 individuals. So these are very large multi-center studies. And this is the placebo group, a low-dose group, and a high-dose group treated for 78 weeks with placebo versus aducanumab. The primary outcome was the clinical dementia rating score, some of the boxes, which is a cognitive functional measure. And this was an example of a successful phase 3 clinical trial meeting its pre-specified primary outcome of the CDR, some of the boxes in the high-dose group with a highly significant p-value, meaning that there was decline in the placebo group, but much less decline in the treatment groups compared to the placebo. So this was a successful clinical trial. Again, the MMSE score, 30 points being normal, losing 3 points, 3.3 points during the 78 weeks of the study in the placebo group, and much less decline, especially in the high dose group. And if we look at this activities of daily living scale, decline in the placebo group, and a 40% reduction in the amount of decline in the high dose treatment group compared to the placebo. So particularly for the high dose group, seemed to be beneficial, meeting its pre-specified primary outcome, and therefore a successful clinical trial. There were also supportive evidence from biomarkers. For example, this is CSF phospho-tau and CSF total tau. This was the placebo group versus low dose and high dose. So you can see a reduction in phospho-tau and spinal fluid as a result of treatment compared to placebo. This would suggest that there was less neuronal death and spilling of tau and phospho-tau into the spinal fluid, and also similar results for the total tau. You can see, though, that these are relatively low numbers because the spinal taps were optional and most individuals elected not to have spinal taps. But of these individuals who did, and we measured phospho-tau and total tau, seemed to be supportive, a positive outcome as in the spinal fluid biomarkers. The problem was this other phase 3 clinical trial called ENGAGE, which was a very similar design, again with more than 500 individuals in each arm of the trial. Here, the clinical dementia rating scale, no difference with a p-value of 0.8. So this was clearly a negative trial. And the question is why and what happened and how can you do 2 large phase 3 clinical trials with 1 positive result and 1 negative result? One difference is that this started slightly later and there was a dose escalation during the trial. So there were fewer patients who got the highest dose in this trial compared to the other trial. But I'm not sure if that's really the explanation or not. No one really knows, of course. And a little bit of supportive evidence in the phospho-tau and total tau, a little bit more messy here. Again, low numbers, but perhaps some supportive evidence of tau and phospho-tau, even though the clinical outcome was negative. What was clear was that in both studies, there was clearance of the amyloid by amyloid PET. So here's the increase in amyloid in the placebo group, the EMERGE study, and the ENGAGE study, versus a decline in the amyloid burden in both the low dose and the high dose group in both clinical trials. So this was a very clear positive outcome, a biomarker outcome, with a reduction in the amyloid burden in both clinical trials. We also did some exploratory tau PET scans with this new tau PET ligand, and this was also supportive. This is a placebo group, increased tau versus decreased tau by tau PET scan in the treatment group compared to the placebo. And if you plot high dose versus low dose versus placebo, you can see that there's a decline in the tau PET signal as a result of the Aducanumab treatment. So again, these are the new tau PET scans that we're starting to also use as outcomes for clinical trials, and this also is supportive of a positive biomarker benefit or outcome from the Aducanumab treatment. These are what the tau PET scans look like. These are individuals who got placebo, baseline versus 78 weeks. So you can see increased tau deposition here, medial temporal lobe, entorhinal cortex, hippocampus. There's another patient, increased deposition, another patient, increased deposition, versus those who got high-dose aducanumab had decreased tau signal by tau PET scan. So this patient, this patient, this patient. So these are what are called representative patients with increased tau PET in placebo and decreased tau PET in the aducanumab treatment group. And as far as adverse events, the major adverse event was ARIA-E, amyloid-related imaging abnormality E. This was up to 34% in the high-dose group and 35% in the other study. So this was a fairly frequent side effect of the aducanumab treatment. Most of this was asymptomatic. Most was reversible, even with continued treatment. But there were cases where it was symptomatic, and the most common symptom was headache in 19% in the treatment group compared to 15% in the placebo group, and also 17 and 20% treatment groups compared to 15% in placebo group. There was also a little bit more of ARIA-H. These are microhemorrhages, sort of pinpoint hemorrhages detectable by MRI scan. They're too small to really make to be of any clinical significance, but they are detectable by MRI scan, and they were more common in the treatment group compared to the placebo group. So both ARIA-E and ARIA-H as the major side effect. If the ARIA-H is near the surface of the brain, it's called superficial siderosis, also elevated. So these are the major concerns, major side effects of the drug. And this is the clinical dementia rating scale, decline in the placebo with less decline in the treatment group. This was the successful study, and this was the not successful study, so no significant differences between treatment and placebo. Again, reduction of amyloid PET in both studies, but if you look in a subset of patients from the phase 3 clinical trial, which was not successful. So this is a post hoc subgroup analysis, which is not to be used for FDA approval, or really for analysis. Then in that case, if you take out the group that got the highest dose for the longest time, also had a significant benefit, and similar to that found with the eMERGE study. And you can see the numbers of individuals here in this study. So again, this is post hoc subgroup analysis of the phase 3 clinical trial, which did not meet its primary pre-specified outcome. But those who got the highest dose for the longest time looked like it did have a reduction in clinical decline compared to placebo. So summary of aducanumab. This study was terminated based on a futility analysis in March, but then when they looked at the larger subset, the eMERGE study was successful, meeting its pre-specified primary outcome, as well as secondary endpoints such as the biomarker outcomes, reduced clinical decline. The ENGAGE study was not successful, did not reduce clinical decline, but a post-hoc subgroup analysis exposed to the high dose was supportive of the positive findings from the successful study. A lot of the biomarkers were indicated potentially disease-modifying benefits for the patients compared to placebo. The major side effect of concern, and probably the dose-limiting side effect, is ARIA-E, and the symptom most commonly caused was headache. And so the company is planning to bring back those individuals from the phase 2 and phase 3 clinical trials to continue treatment, open label treatment, to collect additional safety data on aducanumab. And we hope to start bringing individuals back as soon as we can get back to seeing patients face to face. And as a result of this data, the company is submitting a new drug application to the FDA later this year. So does anyone have any questions, or does anyone have an opinion or an impression about whether this drug should be approved or not?
So there's a question in the chat box.
How to explain the increase in total in the drug group in emerge, increase in total. Oh, in tau. Let's go back. Is it tau in spinal fluid or tau in PET? Oh, spinal fluid, okay. Oh, so this is change from baseline. So this is a reduction in phospho-tau compared to placebo. So this is the amount of change found in placebo, and there was less phospho-tau in the treatment groups. And similar to what we see in the PET scan, there was more tau tangle accumulation in the placebo, but less tau tangle accumulation in the treatment group. So would anyone be in favor of approving this or not approving this, or what, does anyone have a prediction on what the FDA may rule? Catherine Conant would approve. Anyone else have an opinion? John Partridge leaning to no. Sarah, I think a tiebreaker is needed. So one yes, one no, one maybe. And I think my opinion is Ken Lynn says no. I think right now it's kind of 50-50. If you are a true scientist requiring medical scientific data, you would say no, because the traditional criteria has been 2 successful phase 3 clinical trials meeting pre-specified primary outcome. And so we don't have that from these 2 studies. On the other hand, there are politics that enter into the picture, and the FDA has been been in a very approving mood lately. But there may also be some sort of compromise decision. So instead of just approving for prescription use or not approving, there may be some sort of conditional approval requiring further studies or another phase 3 clinical trial. Another phase 3 clinical trial would take another 3 to 5 years and another at least $100 million to complete. It will be interesting to see what the FDA rules. Is the cognitive decline and recovery in the subject? Yes, so that individual was included in the analysis. The patient that we had that had the severe ARIA reaction. Obviously, the drug was discontinued, but the analysis was intent to treat. So this person was definitely included in the analysis. Patients may want it, but then many were clamoring for hydroxychloroquine before any trial. Absolutely, I don't think there's really any good evidence for hydroxychloroquine either. But I know a lot of individuals are looking for that. So that's the Aducanumab story. It's been a very roller coaster ride, as you can see, and which patients may develop ARIA. So the ARIA, the risk of ARIA increases with dose. So the higher the dose, the higher the risk of ARIA-E and ARIA-H. Any individual who's ApoE4 positive has a higher risk of ARIA. And if it does develop, it usually happens within the first 3 or first 5 infusions, sort of like an allergic reaction. So these are monthly IV infusions. And the ARIA usually occurs after, during the first few infusions. So if you can get past the first few and you don't have ARIA, then you're probably fairly safe that you will not have it. So that's the end of Aducanumab for now. We're going to restart dosing our patients who are in the phase 3 clinical trial, phase 2 clinical trials at Georgetown, and then we're waiting to see what the FDA may rule on these results. Obviously, they will have a lot more data than we do, And all of this data that I've shown you on Aducanumab is unpublished data, but it is publicly available on the web. So in addition to doing any peripheral side effects, the only peripheral side effects are just from having an IV every month. There's really no other significant side effects that were found from the ARIA, but there may be some local irritation and reactions from the monthly IV infusion. I'm going to talk a little bit about nilotinib for Alzheimer's. So in addition to all of the large multi-center phase 3 clinical trials that we're doing for Alzheimer's, we are also trying to develop our own strategies. And so this was a single center, single site phase 2 trial done only at Georgetown. And this is based on the work of Dr. Musa's lab, who discovered these effects of tyrosine kinase inhibitors on various transgenic mouse models of neurodegenerative disease, including synuclein transgenics for Parkinson's models, amyloid transgenics for Alzheimer models, TDP43 transgenics for frontotemporal dementia and ALS models. And here's one of Dr. Musa's paper from Dr. Musa's lab. These are transgenic APP mice. So you can see these brown dots here, the amyloid plaque deposition in this mouse model of a placebo-treated mouse. And here's the thioflavin green fluorescence of the amyloid plaques in this placebo-treated mouse. And compared to a nilotinib-treated mouse, which has much less of these amyloid deposits and no green immunofluorescence. This is another tyrosine kinase inhibitor called bosutinib. And again, with the transgenic Alzheimer mouse model, comparing the placebo to the treated mouse, much less in the way of amyloid plaques, as shown here, the green fluorescence in the placebo versus treated mouse. And this can be quantified, the plaque burden of the cortex of these mouse models. So here's the transgenic mouse versus bosutinib treated mouse, and this is also seen with intraneuronal or intracellular A-beta shown here. So not only removing the extracellular amyloid plaques, but the intraneuronal A-beta. So based on all this pioneering work from Dr. Musa's lab, we started a phase 2 clinical trial for Alzheimer's disease, mild to moderate Alzheimer's disease. We assessed 117 and randomized 37, 1-to-1 placebo versus nilotinib. We ended up after allocation and some dropouts with 20 in the treated group compared to 17, 20 in the placebo group compared to 17 in the nilotinib group. So this was a small study, so single site phase 2. And this was the demographics of the individuals, 20 versus 17 in the two groups, 69 versus 72 years old. And the mini mental state score at baseline 19.2 versus 19.8, so fairly equivalent CSF A-beta at baseline. So these had to be biomarker confirmed Alzheimer patients, either with a low CSF A-beta 42 or a positive amyloid PET scan or both. And we also did our ApoE analysis on these individuals. As far as adverse events, potential adverse events were EKG changes. So there were lots of EKGs conducted during this study, but there were no significant risks found on the EKG. The other potential side effect is GI, including diarrhea, and there was 15% diarrhea in the placebo versus 41% in the treated group, so there were more GI side effects. There were also a few individuals who had elevated LFTs and amylase. I'm not sure if this was a lab anomaly or true elevations. That's something to watch in future studies, but really the only significant AE that was found was more mood swings in the higher dose, at the higher dose. So these individuals were given placebo or 150 milligram of nilotinib for 6 months, followed by 300 milligram nilotinib for 6 months. So once we switched to the 300 at the 6 months, we found more mood swings, irritability, agitation, in the treated group compared to the placebo group. So this was really the only significant adverse event that was noted. As far as serious adverse events, there were 5 in the placebo group and none in the nilotinib group. Fortunately, I think this was just good luck. But you can see there were 3 individuals who had 5 SAEs in the placebo group. And then when we started looking at biomarkers, you can see, for example, hippocampal volume is measured by MRI scan. There was less atrophy found in the treatment compared to the placebo in hippocampal volume. There were significant declines in CSF A-beta 40 and A-beta 42 in treatment compared to placebo. Looking at the total tau, nothing significant, but phospho-tau, some benefit in phospho-tau levels in spinal fluid. And also perhaps indication of higher dopamine in brain, because DOPAC is a metabolite of dopamine in treatment compared to placebo. But no effect on ABL or phospho-ABL. So one hypothesis was that this drug is inhibiting ABL, which is a tyrosine kinase, but we found no evidence for that in CSF by these looking for phosphotyrosine. As far as clinical outcomes, this study was grossly underpowered for any sort of clinical benefit or clinical outcome, and we knew that from the beginning. You could tell from, for example, from the phase 3 clinical trials that you really need at least 500 individuals per arm to get a significant difference on clinical or cognitive outcomes for Alzheimer clinical trials. So obviously with these small numbers, we're not expecting to find anything significant, and we did not find anything significant in clinical outcomes. So the placebo versus treatment group, although there's a slight indication that there may be less decline in MMSE in treatment compared to placebo. And then all of these are indicating worsening during the course of the study. If you look at one particular test, which is time to completion of one of the tests in the Alzheimer's disease assessment scale, there was perhaps a significant benefit with less time to completing in the treatment group compared to the placebo. But this is just one task out of the ADAS. And this is a clinical dementia rating scale, activities of daily living scale, neuropsychiatric inventory scale, but basically really nothing significant happening as we would anticipate. But probably the most exciting outcome from this clinical trial was the results of the PET scans, amyloid PET scans. And here's the placebo group, an increase in amyloid burden during the 12 months of treatment with nilotinib compared to the drug treatment with a decrease in amyloid burden and a significant difference in the frontal lobe burden of amyloid plaque deposition in nilotinib treated compared to placebo treated. Similar trends, but not significant in temporal lobe as well as in total brain. So this increase in amyloid burden in placebo and a decrease in the treatment group. So here's an individual at baseline. Oops. Here's an individual at baseline, cerebellum versus frontal versus temporal. And this is baseline, and 12 months later, you can see increase in amyloid deposition compared to a nilotinib treated individual. If you compare frontal regions here compared to here, there seems to be a reduction in the amyloid burden in this individual. And so I think this is a real effect. This is the first oral drug which lowers amyloid burden in Alzheimer's disease patients. Now, this has been seen in several antibody studies, including Aducanumab, but has not been found with any oral drug until this study. So just to summarize some of the tyrosine kinase and nilotinib studies, I won't mention the Parkinson's study, which preceded the Alzheimer trial, But similar to the Parkinson's trial, nilotinib seems to be safe and well tolerated in elderly Alzheimer's patients, reduces CSF phospho-tau, beta 42, amyloid burden, and hippocampal volume loss, so supportive biomarker evidence of a potential disease-modifying effect. And obviously, we'd like to extend and expand this strategy of not just nilotinib, but other tyrosine kinase inhibitors, including some novel compounds that Dr. Moose's lab has under development. And not only for Parkinson's disease and Alzheimer's disease, but there's also trials now underway for Lewy body dementia, which also has the alpha-synuclein aggregates causing not just Parkinsonism, but also dementia. And we would like to move on with either phase 2 or hopefully phase 3, larger multi-center clinical trials for Alzheimer's disease and Parkinson's disease in the planning stages. But of course, the major challenge is getting funding for these larger studies. The phase 2 study that we conducted for Alzheimer's, I think the cost was at least $3 million, and a large amount of that cost was in buying the drug from the pharmacy because the company declined to donate the drug. So obviously a much larger phase 3 trial is going to cost quite a large amount, and so it will be a challenge to get the funding for a larger study. So does anyone have any comments about the nilotinib study? Has the apparent disconnect going right down clinical cause of rethinking? So there's this disconnect. As well as, this amyloid hypothesis is still a hypothesis. We haven't proven it. We won't prove it or support it until we have some sort of amyloid reducing or amyloid targeting drug that is shown to be efficacious, which you could argue has not really happened yet. There certainly is this disconnect between amyloid burden and cognition. You can even see that on when biomarkers become abnormal. I think our thinking is that the amyloid drives other pathologies, including the tau and tangles. And this is supported by finding effects on tau and anti-amyloid treatment strategies. So there certainly is this disconnect, and there's a lot of debate and rethinking about the whole amyloid hypothesis, because there have been so many negative trials that I've not mentioned in the past. But I think the amyloid hypothesis is still our sort of best hypothesis so far, and the major support comes from the genetics and the genetic mutations and the genetic resistance genes that have been found. This have some influence on the amyloid generation and production and deposition in brain, but it is still a working hypothesis, and there's a lot of debate about whether it's correct or not. Reductions in A-beta and phospho-tau comparable between nilotinib and aducanumab. Yes, for the most part they are, but there were relatively small numbers for both studies. Remember, the aducanumab only had a few individuals who elected for spinal tap studies. Nilotinib, we also had relatively low numbers, but the order of magnitude was similar in both studies, but there's a caveat in that both studies had relatively small numbers. So I don't think we really know yet. We need larger numbers. Why would the company not want to fund a nilotinib study? That's a great question. The company did donate drug for the Parkinson's trial because there was a previously published phase 1 study by Dr. Moose and Dr. Pagan. Since there was no published study for Alzheimer's disease, they did not donate drug for Alzheimer's disease. And we don't really know why they didn't get more excited or enthusiastic about this approach, but we hope that as more data is being collected, not just here, but elsewhere, on this tyrosine kinase strategy for neurodegeneration, the company will become more interested. And I think the scientists were more interested than the business people at Novartis, which is the manufacturer of nilotinib. One issue is that the nilotinib is still under patent protection for leukemia, and there are still 2 to 3 years left on the patent, and the company is profiting or is selling nilotinib at about $1 billion per year currently. And I think there was some concern as to whether we may find some side effect of nilotinib which might impact on their sales of nilotinib for leukemia. So we don't really know why the company was not more interested, but we hope to get them more interested in the future. And if not, if they don't want to fund a clinical trial, at least they would donate drug and matching placebo for future studies. But this is all remains to be seen what we can accomplish next. Anything else about tyrosine kinase inhibitors? Preclinical data on nilotinib in animal models to delay MCI or dementia pre-treatment idea. Yeah, that's a good idea. Most of our studies with the animal models have not been prevention studies, they've been more treatment studies. And we would like to move more into prevention studies. So, there's obviously a lot more work to be done with the animal models in addition to human studies. But once we can find an effect on treatment, we would like to move on to prevention. But prevention typically requires larger numbers and longer amount of time, meaning more expense. So it is something for future directions. So just some of the trends that have been happening in our Alzheimer's research. Obviously, we're developing biomarkers. And I've shown you some of those: glucose PET, amyloid PET, tau tangle PET, and perhaps also inflammatory PET ligands that are under development. There's a lot of interest in proteomics and other omics in bodily fluids, including spinal fluid, particularly A-beta, phospho-tau, and tau and neurofilament light in CSF. And there's obviously a huge amount of interest in a blood test for Alzheimer's disease. And we're getting pretty close with A-beta and tau measurements in blood, very sensitive measures to detect these proteins in blood. Not quite ready for prime time, perhaps may be useful in the future as a screening test for those who are on the trajectory to get MCI and Alzheimer's. We're moving earlier in the Alzheimer's spectrum, away from Alzheimer's and towards MCI and prodromal Alzheimer's based on biomarker studies, doing more MCI trials. In addition to drug trials, there are a lot of lifestyle interventions that are being investigated, including exercise. And obviously trying to move more towards prevention. So we're starting to do prevention trials for individuals who are at higher risk, either ApoE4 positive or amyloid PET scan positive, or other biomarkers that indicate someone is at higher risk. In order to find these normal, healthy, older individuals, we're doing more web-based screening and recruitment. And one positive thing that's happened in recent years has been a quadrupling of NIH funding for Alzheimer trials. So this is certainly encouraging to those in the field and hopefully encouraging to those who would like to get into the field of Alzheimer's research. In the memory disorders program, we have several studies that are in progress or will start soon. These are the list of studies that have no drug whatsoever. These are just biomarker discovery and validation. And this is the Alzheimer's Disease Neuroimaging Initiative, including the DoD, ADNI, which is enrolling veterans with PTSD or TBI, looking for their Alzheimer risk over time, looking for early language changes in MCI and Alzheimer's with a collaborator at Marymount. We're starting our own DC longitudinal study of aging and specimen collection to collect blood and longitudinal clinical data, and to find a local cohort who may be interested and eligible in other clinical trials. This is a trial coming soon to discover new genes linked to early onset Alzheimer's disease, people less than 65. And these are people who we're following longitudinally with a negative amyloid PET scan to see what happens to their cognition over time. And this is another effort to find interested and eligible individuals who are maybe interested in joining a clinical trial or a prevention trial in the future. trying to increase our efficiency of recruitment. These are some of our drug trials underway for either MCI, prodromal Alzheimer's, or Alzheimer's, nicotine patch, BAN2401, which is another anti-amyloid monoclonal, another one, gantenerumab, a study coming soon, metformin for Alzheimer's, based on a link with diabetes and Alzheimer's disease. An anti-tau antibody, we're also studying this as an approach, not just anti-amyloid antibodies, but anti-tau, tangle antibodies. We're continuing with Aducanumab, another new study, another anti-amyloid antibody. So each one of these antibodies, of course, is a monoclonal with different epitopes, different affinities that binds to different conformations of the amyloid, and so that may have different effects and potencies. And there are more studies coming. We're also doing prevention trials, particularly the A4 study, which is another anti-amyloid solanezumab. So this is a 7-year study, finding people who have a positive amyloid PET scan and treating them just, while they're still cognitively normal, to see if we can prevent cognitive decline. And there are more prevention trials on the way. One way to help us find healthy, normal, older individuals interested in research is by this web site, which is called aptwebstudy.org. Anyone in the US, anyone who's over 50 can log in and register at this website and learn about how to keep your brain healthy with aging and prevent Alzheimer's disease, various lifestyle and other interventions. It will also do brain games or tracking your cognitive performance. And if you would like, you can repeat this testing every 3 months. And if the website detects a decline in your memory or cognition, it will send you information about a local research center where you could learn about opportunities for research and opportunities for prevention trials if you are so inclined. So this is just another way to try to contact healthy, older, at-risk individuals for prevention trials. So again, here's our, everyone in the Memory Disorders Program, and thank you to all of you and everyone in the Clinical Research Unit and the GUTS who are our collaborators. I have some time for maybe 1 minute for questions. Would you expect tyrosine kinase inhibitors have similar impact? Other tyrosine, let's see, willing to fund clinical trial. Yeah, there's, I think there are about 25 or so tyrosine kinase inhibitors on the market already. I don't think the blood-brain barrier penetration is very good for most of them. There are a few that get into the brain. I think Dr. Musa has done some research along those lines. Nilotinib and bozitinib are, I think nilotinib is one of the ones that gets into the brain the best. They're obviously not designed for Alzheimer's disease. This is a repurposing study. They're designed for leukemia. So I think there's a lot of room to study other tyrosine kinase inhibitors, to develop novel molecules of tyrosine kinase inhibitors, and to target tyrosine kinase inhibitors to those that may be most involved with Alzheimer's pathologies or other neurodegenerative diseases and Parkinson's disease. So I think there's still a lot of room to go in developing this strategy, this tyrosine kinase inhibitor for neurodegeneration. Any other questions?
Not seeing any. I'd like to thank you, Scott, for just an extraordinary and sweeping review of a huge clinical research subject, and one with huge national clinical impact. Thank you for sharing that work with us.
Yeah, sure. Thanks for having me. I look forward to seeing everyone in person one day.
From your mouth. Bye, everyone. Thank you for joining us.
Thank you. Bye.